tumor. It is related to Peace-Jaeger syndrome, and the mutation for Peace-Jaeger syndrome is STK11. You need to remember it for the exam purpose. Sertoli cell tumor, as I mentioned, the counterpart of granulosa cell tumor. So receive the signal from FSH and secretes estrogen, and also secretes inhibin as a negative feedback. But the symptom here, I don't know why it's still related to some estrogen manifestation, not just estrogen. But I think it may make sense because in normal ovary, you could not find this Sertoli cell. So maybe they become tumor, they can have different kind of behavior. Then the growth picture also similar. Yellowish cutting surface, it also can become a big one. Mean average size is 8 centimeter. The microscopic feature, I think you just need to remember the tubular structure because this is very characteristic. And the cell is also kind of characteristic but not really distinct. You can see we kind of deeply eosinophilic cytoplasm with this columnar cell, but forming tubule, that is really characteristic. See, forming this compact tubule, and then the tumor cells forming this next structure separated by this fibrous septate. High power, you can see this cell is forming this tubule with this empty space as a cytoplasm. You can still see the tube lumen just squeezed by the tumor cell and then forming this tubule. You need to pay attention, they can also form this endometrioid gland, endometrioid carcinoma-like gland, but still this is still tubule. But the immunostaining can really help us. The immunostaining also express inhibin, SF1, carotene, WT1. It also related to DICER mutation. What is the function of DICER gene? This is a really important gene for the expression of microRNA, and microRNA is a really important regulator to suppress the gene expression. So you can imagine if you lose this microRNA regulation system in your body, and then tumor just lose control of some oncogene, and then it is really important gene. And also this STK11, aka LKB1 mutation in Peace-Jaeger syndrome. The differential diagnosis, always remember we have this mixed cell tumor, Sertoli cell and Leydig cell tumor. So if you find abundant Leydig cell component, you may really have suspicion it's not just Sertoli cell tumor. It should be a mixed Sertoli Leydig cell tumor. I will talk about Leydig cell tumor more later. And then melanin is the specific marker for Leydig cell in this entity. And also endometrioid carcinoma. In this entity, we should find some intraluminal or intracytoplasmic mucin, and also squamous differentiation. EMA PAX-A, our epithelial marker, our organ-specific marker, relatively PAX-A, and this sex chromosomal cell marker inhibin and SF1 negative. Steroid cell tumor could be in the differential. I will talk more about it later, but you just need to remember steroid cell tumor and Leydig cell tumor actually they are the same. But if you think this is a Leydig cell tumor, but you could not find Reiki crystal, you should diagnose it as steroid cell tumor, NOS. And also this germinoma, because when you remember this Sertoli cell tumor can forming nest, and then this tumor nest is surrounded by fibrotic septa. This is just a feature of this germinoma, aka their counterpart seminoma in the testicle. The only difference is that in this fibrous septa, you can find lymphocytes, and then the tumor next, usually they do not form any tubule structure. And also immunostaining for germ cell marker is really characteristic. Okay,